This time we're going to study respirometers, and respirometers basically are to measure the rate of oxygen uptake. Rate of oxygen uptake. Now if you remember your O-level equation, we said glucose plus oxygen gives you carbon dioxide plus water. So if we know how much oxygen is being taken up per unit time, like if when an organism is taking up 100 molecules of oxygen in a minute, or another organism is taking up 200 in a minute, so we would know the rate of oxygen uptake. You see, rate is always something to do with the time. So now looking at, I've just got these two respirometers there for you. The basic ingredient for a respirometer, number one, has to be a place where you put the organisms. So a chamber where you place the organisms. And then you have to have a chemical which absorbs CO2. Now in this situation here it is soda lime, and here again they have soda lime. So you have a place where the CO2 is absorbed. CO2 is absorbed. So you have to have something which is going to absorb the CO2, because you see in certain equations, like we have 6 O2, 6 CO2, so there's going to be no change in pressure. So because if 6 oxygen is being taken up, 6 carbon dioxide is produced, so we are not going to see any change. How are we going to measure the change? This is the reason why we have to put a chemical. And number 3, we have to have some sort of capillary tube. We have to. Why do we have to have a capillary tube? Now that is something which you are not very clear about. A capillary tube means a tube with a very narrow bore. Now you can't have a tube which is a wider bore or a wider lumen. The reason is because we want to see the movement of that colored liquid. This colored liquid or this manometer fluid which we have here. So if you have a narrow tube, even a small movement can be easily registered. So if you had a very thick tube like this one here, this is a very thick tube. Now if a lot of oxygen is taken up, only then some sort of a dye will move. Now this just I'm trying to explain to you is, having a tube with a wider lumen, why is that not really justified? And we use a very tube with a very thin lumen, because we want to see the movement of the colored liquid. So there's a colored liquid here, there's a manometer fluid, and then this is going to move towards the organisms. In this case it is going to move this way. So again I'm just going to do a recap. A respirometer is an instrument which measures the rate of oxygen uptake. It has to have 3 things. Number 1, a place or a chamber or a place where you cavity, like in a test tube or in a syringe. Could be anything. Basically 3 चीजें होनी चाहिए. So one where you place the organisms, and one where you place a chemical. The chemical should not come in contact with the organisms because it could be toxic or it could be harmful. And number 3, a capillary tube which is going to measure, which is going to have a dye or a colored liquid in it, and which is going to move, and we're going to measure the movement. We're going to measure the movement कि कितना move किया. Does it move 20 millimeter? Did it move 30 millimeter? Similarly here also, there will be a scale here which is going to measure how much this dye or this manometer fluid is going to move. So respirometers are basically, you can have any type, but they have to have these 3 ingredients which I have just, so 3 things which are important for a respirometer, and that is what you have to understand. Now looking at this diagram that I have found for you all, we've got 2 test tubes. In this we have 2 test tubes. Why do we have 2 test tubes? Because one has the living organisms, like the respiring organisms, which give off CO2 and absorb oxygen. And then you have these soda lime pellets. Then you have the same one on this side. And these are going to absorb the carbon dioxide. So number 1, the respiring organisms give off carbon dioxide and absorb oxygen. Number 2, carbon dioxide is absorbed by the soda lime. Number 3, the manometer fluid moves to the right due to the fall in pressure as the volume is reduced by the uptake of oxygen. ये pressure वाली कहानी everybody must understand. Pressure difference create होता है इसलिए वो move करता है fluid. Now this tube is a control, but it has been labeled as a thermobarometer. And in this we have placed glass beads. I asked you this question the other day in class. What has to be kept constant is the volume. So the volume must be contained constant because they occupy space, the living organisms and the glass beads. So any change in pressure or temperature during the experiment affects the respirometer tube and the thermobarometer tube equally. क्योंकि देखें अगर we didn't have this tube, then just imagine you didn't have this part. So any pressure exerted on this tube here, air pressure from the outside, would make the U-tube manometer, sort of the liquid, moving towards that side. So what you have to understand is, why did we have this on this side? The reason why we had this on this side is because this was number 1 a control, and number 2 to equalize pressure. Equalize pressure. Now number 4, after a fixed time the syringe is adjusted to level the fluid in the two arms of the manometer and the volume of oxygen absorbed is read off on the syringe. You see, what you have to remember is this is the length of a column, and the formula for that would be pi r square h. If we knew the diameter, then we'd calculate the radius and we would then have the volume. This is the length of a column. ये जो move किया ये die, अगर ये इधर ऊपर को move करेगी, कितना move करी है? 20 millimeter move किया. 20 millimeter is the length of a column. ये हमें कुछ नहीं बता रहा. So what you have to understand is that after a fixed time, the syringe is adjusted to level the fluid in the two arms of the manometer and the volume of oxygen absorbed is read off in the syringe. So this is how you use this formula. So you have to understand is that we are measuring the volume of oxygen used per unit time. And then of course if we were using whatever organisms, like we're using seeds or we are using invertebrates, then we would have, if we were comparing, then the same mass would be used. And we would then see how the rate was different. You're not supposed to know whether the rate is more or less. That is not what you're supposed to be really worrying about. Now coming to the RQ. RQ is respiratory quotient. So when we talk of the RQ, we are expecting you to know this formula, which is volume of CO2 over volume of O2. So we'll give you an equation. And in that equation, we usually give you the equation of C6H12O6 plus C6H12O6 plus 6O2. And this gives you 6CO2. Now you know 6O2, 6CO2 plus water तो कर लें. अब इसका RQ simply क्या है? ये वाला 6 over this 6, so is equal to 1. That is why we say carbohydrates have an RQ. Carbohydrates, you remember glucose is a carbohydrate. So carbohydrates have an RQ of 1 because it's 6 over 6. Now the others which you have to remember, which also can be given to you in a formula, is fats have 0.7. Now it's very easy to, I usually remember it very easily. I say okay, ये F of fats, तो इसको उल्टा कर दे तो ये 7 हो जाता है. And then P for protein, P को उल्टा कर दे तो 9 हो जाता है. I mean mirror image, also उल्टा. So carbohydrates 1, fats 0.7 and proteins 0.9. So they're all less than 1. But then when we do anaerobic respiration, well, we have a problem here. You know, glucose, there's no oxygen. Oxygen तो है ही नहीं. यहाँ पर तो हमने oxygen लिखी थी. ऑक्सीजन तो है ही नहीं, gives you whatever 2 CO2 or 5 CO2 or 10 CO2, that's immaterial. But when we look at the equation for RQ is CO2 over O2, so it's 2 over 0, that's infinity. So what does RQ tell you? RQ tells you two things. Number one, it tells you what is the respiratory substrate. If it's 1, you know it's a carbohydrate. If you know it's 0.7, you know it's a fat. If you know it's a protein, it's 0.9. Number 2, it tells you, is it aerobic or anaerobic? Is it aerobic respiration or anaerobic respiration? Why would it tell you that? Number 1, if it is 1 or less than 1. If it is 1 or less than 1, then it has to be aerobic. But if it is more than 1, because you see now we are looking at this figure, infinity. So if it's more than 1, that means there is some aerobic and some anaerobic respiration taking place. So this is what we needed to understand about RQ. The RQ is the respiratory quotient. It is the volume of CO2 over volume of O2. The equation you must know these stand figures which I have given you for carbohydrate 1, fats 0.7 and proteins 0.9. Now how do we measure the RQ using a respirometer? Now as I told you, RQ is CO2 over O2, right? So when we did this experiment and we saw the movement, we got a value. Say you got a value of 6. So that tells you 6 is the oxygen uptake. Again, I'll explain it again. When we set up this respirometer, we set it up and we sort of got one reading, then we reset the apparatus, we got a second and third, and then we got a mean reading, and that was 6. Volume of oxygen taken up for the germinating seeds or anything else, invertebrates. But the trouble is, we don't have this figure. So how do we get this figure? This is a big problem कि हमारे पास तो ये figure है ही नहीं. अब इस figure के लिए हम क्या करते हैं? हम इसी apparatus को इसी तरह रहने देते हैं, same organism, same everything else, except that we remove the soda lime. We remove this soda lime. Now when we have the soda lime, you know that the CO2 is being absorbed by the soda lime, and somebody might say, okay miss, let's measure the mass of the soda lime. If that is increased, that will tell us the CO2 uptake. That's correct in a way, but it's very negligible. So what we do is that we reset the apparatus, everything goes back, the manometer fluid goes back to the 0 point, they're both at the same level, and then we use the same seeds or whatever is present, and we've removed the soda lime, and now we see there is no movement. Now we see that there is no movement. Now what is this telling you? It means क्या हुआ है? It means उसने 6 oxygen लिया है और 6 CO2 produce किया है. So that is how we figure out. But remember one thing, we had to have this figure beforehand. We had to have the oxygen uptake figure beforehand. Then what did you do? So same apparatus, everything same, but remove the soda lime. Okay, first the apparatus with the soda lime gives you this figure. Then the apparatus without the soda lime tells you there is no movement. So if there is no movement, it means कि इसने 6 oxygen लिया इस organisms ने और 6 CO2 produce किया, इसलिए movement कुछ नहीं हो रही. So the value is 1. Now of course, this is now the different values that we can have. Now the different values that I talked to you about is 6 over 9 or 9 over 6. And there's a reason why we talk about these. So this means this is CO2 and this is O2. This is CO2 and this is O2. Now just imagine what is going to happen. So if we have, this is O2, O2 means 9 O2 ऊपर गया है, उसने लिया है, और 6 CO2 प्रोड्यूस हुआ है. So ये 9 है और ये 6 है, तो ये +3 पे आ जाएगा. If the other thing has happened, what is the other thing? 6 O2 लिया है उसने और 9 CO2 प्रोड्यूस किया है, तो ये -3 पे चला जाएगा. So So ये दो आपको मैंने कहानियां करनी थी. एक, when it does not change, that is 6/6. Now you know 6/9, what would that result in? 6 divided by 9, 9/6 will be 1 point something, 6/9 will be something. So you do this calculation and you figure out that RQ can vary and how we will change the respirometer. So first we will use the respirometer with the soda lime, then without the soda lime. The same respirometer. If I say, okay, how are we, this comes in your paper 5 a lot. So how are we going to amend the apparatus and measure the RQ using a respirometer? Now the last topic in this chapter is about rice, and rice is a swarm plant, and you can see how, I'm sure you've seen it, we see a lot of paddy fields around us. It's not something that we've not seen. We see a lot of water standing and the plant growing in a lot of water. Now this cannot happen to other crops. They will not survive this. Agar flooding hoti hai, crops mar jate hain. But how is the rice able to survive? So you see, rice is a swarm plant and it has special air spaces in its leaves and its stem, which is called the Aranchyma. Now the Aranchyma, now if you look at a plant, a rice plant, it's got all this water, so the roots will die. The roots start to anaerobically respire. When they respire anaerobically, they're going to produce alcohol, ethanol. Now if there is a lot of ethanol, the plant is going to die. It's toxic for the plant. So what we see is, number one, we see a lot of air spaces. We see a lot of air spaces inside the leaves. The leaves also have air spaces in it, more than the normal leaf. And then the stem also has air. If I take a section of the stem, you see a lot of air spaces in it. So what is going to happen here? The leaves is going to, air is going to enter, then it's going to go through the stem, and then it's going to go to the roots. So air or oxygen entering the leaf is going to go through the stem and enter the roots. But the roots are submerged in water, so they cannot respire aerobically. Now the name of these air spaces you have to know is called the Aranchyma. So the Aranchyma is the part which is going to, you're going to get histology slides on it, and you're going to sort of label that part of in a rice stem or a rice leaf where the Aranchyma is present. Then your book says something which I'm sure when you read it you probably don't get the hang of it. It says that the leaves of the rice plants have these ridges. They're not sort of smooth, they have these ridges. And air is trapped in between the ridges of the underwater leaves. So if there is a water molecule, the water sort of comes around here. This is the water. Now you see the leaves have a hydrophobic corrugated surface that holds a thin layer of air. So this is the layer of air. These leaves have a hydrophobic corrugated surface that holds a thin layer of air in contact with the leaf surface. So this is the leaf. So this is the way how it manages to survive in this, when the roots are all submerged in water. Besides this, they also produce more alcohol dehydrogenase. Now why they produce more of it? Because it will break down the ethanol, and then of course it will not harm the plant, because ethanol is going to harm the plant, it's going to kill the plant. So even when there is not enough oxygen available, they produce more of this enzyme, alcohol dehydrogenase. Baki plants ये नहीं produce कर सकते. So please understand how the rice is adapted for wet environments. Okay, last question, which is an interesting question. When we study muscles, When we study muscles and anaerobic respiration, when you're doing some sort of a very, very vigorous exercise, now why can't we calculate the RQ of this? No RQ can be calculated for muscles. Why? Because CO2 over O2 की कहानी है. Anaerobic respiration, ऑक्सीजन भी नहीं है, CO2 प्रोड्यूस नहीं होती. So no CO2 is produced, there is only lactate produced. So in the lactate pathway or in muscles, there can be no RQ calculation. I can think of a question which would be set on that. Why do we not calculate? Why can we not calculate the RQ of the lactate from a lactate pathway? Okay, ji, thank you.